Imagine opening a laboratory door and seeing an animal that vanished 90 years ago, breathing quietly beneath the lights. It has the striped back, stiff tail, and watchful eyes of the Tasmanian tiger, the thylacine, alive again. But before calling it a resurrection, scientists would face a harder question. Would this creature truly be a thylacine? Because rebuilding an extinct animal is not like printing a body from a perfect genetic file. The file is damaged. The last known captive thylacine died at Hobart Zoo on September 7, 1936, after years of human persecution. Government bounties, habitat pressure, hunting, and fear reduced a unique marsupial predator to photographs, skins, bones, and silence. Yet inside museum cabinets, preserved specimens still contain something more valuable than appearance: fragmented chemical instructions for building life. DNA is often called a blueprint, but ancient DNA resembles a library, shredded, stained, and mixed with pages from elsewhere. Researchers sequenced a thylacine genome from a preserved pouch young, reconstructing much of the animal's genetic instruction book. Later work arranged those fragments into chromosome-scale sequences, using genomes from living marsupials to help restore the missing order. Scientists have even recovered historical RNA from century-old thylacine muscle and skin, revealing traces of genes once active in those tissues. Small dasyurid, such as the fat-tailed dunnart, offers living cells, reproductive biology, and a possible bridge to the extinct predator. Scientists compare the dunnart and thylacine genomes, searching for differences connected to body size, skull shape, fur, metabolism, and development. Then gene editing tools could change selected regions, gradually moving a living marsupial cell toward a thylacine-like genetic design. But identifying a difference is easier than proving what it does. One altered letter may affect several traits at once. Many crucial changes lie outside protein-coding genes, inside regulatory regions controlling when, where, and how strongly development unfolds. The thylacine looked remarkably wolf-like, although marsupials and placental wolves followed separate evolutionary paths for roughly 160 million years. Studies of preserved pouch young show that this resemblance emerged through development, not simply through a handful of obvious genes. Researchers therefore need more than a genome. They need a developmental map explaining how cells become jaws, limbs, organs, and stripes. After editing, scientists must transform cells into reproductive cells or embryos capable of completing every stage without hidden defects. That embryo must implant, form organs, establish maternal exchange, and survive precisely timed chemical signals from its surrogate. A genome from one museum specimen represents one animal, not the full genetic variation once carried across wild populations. Without diversity, a recreated population could be vulnerable to infertility, inherited disorders, new diseases, and changing environmental pressures. Scientists might compare several specimens to recover alternative genetic variants, but every sample contains different gaps, damage, and contamination. Choosing which variants to use would become an act of reconstruction and, inevitably, a series of human decisions. Then comes a deeper problem. An animal is influenced by more than the letters written in its genome. Chemical markers can change how genes behave, and many of those developmental settings are not preserved inside a genome sequence. Microbes living in the gut, skin, and mouth help shape digestion, immunity, and health, yet the original community is gone. Behavior also cannot be downloaded. A predator must learn how to hunt, explore territory, avoid danger, and respond to other animals. No adult thylacine remains to demonstrate those skills, correct mistakes, or provide the social and environmental cues of its species. A hand-reared animal might look historically accurate while behaving in ways that no wild thylacine ever naturally did. And the landscape awaiting it would not be the Tasmania of 1936. Roads, farms, diseases, prey, and climate have changed. Scientists would need to identify secure habitat, sufficient prey, manageable disease risk, and communities willing to coexist with a predator. Releasing an unfamiliar carnivore could alter prey numbers, compete with existing predators, or create consequences that computer models missed. Yet keeping it permanently behind fences would raise another question. Was the species restored or merely recreated for exhibition? The International Union for Conservation of Nature uses the term proxy because a recreated organism may resemble an extinct species without duplicating it. Under that definition, success depends not only on appearance, but also on health, behavior, population performance, and ecological function. Perhaps the goal should not be genetic perfection. Perhaps it should be restoring a lost role without causing new harm. But ecological function is difficult to prove when scientists still debate exactly how thylacines hunted and shaped their environment. There is also the welfare of surrogate animals, whose bodies may carry embryos created through repeated experimental attempts. Every failed pregnancy, malformed embryo, or unhealthy newborn would represent a real cost paid by a living animal. Critics also ask whether enormous resources should revive the dead while many living species approach extinction right now. Supporters answer that the same genomic, reproductive, and biobanking tools could protect threatened marsupials before their populations disappear. Better genome assemblies can reveal harmful inbreeding, preserve rare variants, guide breeding programs, and improve long-term disease management. Stem cell methods, embryo technologies, and artificial developmental systems could become emergency tools for species with almost no breeding animals left. In that sense, the thylacine may function as a demanding test case whose technologies benefit animals that can still be saved. But that promise depends on transparency, independent evidence, strict animal welfare, and conservation work continuing far beyond the laboratory. The safest lesson of de-extinction is not that extinction can be reversed, but that preventing it remains vastly easier. A recreated thylacine would still need years of breeding, veterinary care, behavioral study, habitat preparation, and cautious ecological testing. Its first breath would not finish the project. It would only begin the longest and most uncertain stage. And society would have to decide who owns it, who protects it, where it belongs, and what risks are acceptable.